Everyone knows how expensive medicine is, but very few people know that it takes a long time to make just one medicine. What if we needed to make hundreds of medicines to find the best one to fight a possible pandemic? We'd need a faster, more efficient method. I'm Jennifer Schoborg, a chemical engineer, and I'm working on making medicines faster with cell-free protein synthesis. In the 1970s, insulin was the first protein-based medicine to be made. Since then, we've made changes to the method we've used to make medicines like Herceptin for breast cancer or Lactaid for lactose intolerance. The only thing is, we've kept relying on the same time-consuming step, growing cells. So what are proteins, and how do cells come into play? Proteins are the workhorse of cells. They help cells live and grow and react to their environment. So the cells I'm talking about could be the trillions of cells in your body, or a small yeast cell you use to make your bread. Before cells can use these proteins, they have to make them. So they start off with 20 different amino acids, which are like building blocks for proteins. Then, within the cell, there's complex machinery that's able to put these building blocks into a specific sequence. This sequence determines a protein's structure and function, and the function is what we really like for medicine. About 40 years ago, scientists realized that we can force cells to make the proteins we want for medicine. The problem is, this still takes a long time because we're waiting on cells to grow, so it could be days or weeks before we have the medicine we want. And that's if everything goes well. If the protein's really hard for the cells to make, it could be taxing on the cell, which could make the cell sick and then grow slower, making it longer until we have the protein we want. To get around the problems with cell growth, my lab mates and I use a new method called cell-free protein synthesis. So we take the yeast cells I mentioned earlier, open them up, and take everything inside and put it in a tube in liquid form. What's amazing is that we can add energy, amino acids, and other ingredients, and the machinery still works to make the protein we want, even though it's not within the cell. So this is kind of like a factory. If the cell is the factory and the machinery inside is what we want, the factory is just a building, but we can take the building away and the machinery still works. Now that we no longer depend on living cells, we have a lot more flexibility. One advantage is that we can use conditions that we couldn't use before. So we could use temperatures where yeast cells wouldn't have grown, or add molecules that could have killed the cells but could help make our protein. Another advantage is that we can make lots of proteins at one time. So we could take this plate and make 96 different proteins in each of the different wells. With the millions of naturally occurring proteins and the infinite number of ways we could modify and design them, there should be no shortage of possibilities for new medicines. But cell-free protein synthesis could revolutionize the way we make and discover these medicines. So, who knows? Someday, we could use this new method to nip the next pandemic in the bud.